Imagine stepping into a state-of-the-art medical research facility.
Right, the real cutting-edge stuff.
Exactly. You're looking at the front lines of the war on cancer. You see the white lab coats, the sterile benches, millions of dollars of equipment.
It's an intimidating environment.
It really is.
Yeah.
And there's this underlying expectation we all share, Like, if these brilliant scientists can cure a disease in that perfectly controlled lab environment, well, it's eventually going to translate to human patients in a clinic.
You would certainly hope so.
But what if there is a massive glaring blind spot built right into the very foundation of how we test these therapies?
And it is a blind spot that, quite frankly, has cost us decades of potential progress, especially when you look at something as devastating as lung cancer.
Yeah, and that is exactly what we are unpacking in this custom-tailored deep dive designed specifically for you. We're looking at a glaring paradox today.
It's a big one.
Think about this reality. Lung cancer is a disease that predominantly strikes older adults. I mean, the median age of diagnosis is around 70 years old.
Right.
Yet, if you look at almost all the foundational laboratory research, like the mouse models we rely on to understand the mechanics of the disease and to test new cures, those experiments are conducted on mice that are only 2 to 3 months old.
Which, if you convert that to human biological time, means we are trying to understand and cure a 70-year-old's disease by studying the cellular equivalent of human teenagers.
It sounds absurd when you say it out loud.
It really does.
So, our mission today is to explore a truly groundbreaking March 2026 paper published in the journal Nature. It was led by a researcher named Patel and their team.
A fantastic team, by the way.
Absolutely. And this paper reveals exactly how the physiological process of aging fundamentally reprograms the way lung cancer behaves and evolves.
It changes everything we thought we knew.
It really does. But before we get into the heavy cellular mechanics, let me just float a thought out there for you listening. If you picture an aging body, you picture an immune system that is generally weaker, right?
Sure, less resilient cells.
Exactly. So wouldn't logic dictate that a tumor would just grow much larger and much faster in that older environment?
Well, what's fascinating here is that the actual biology is completely counterintuitive.
Oh.
Yeah. The most surprising discovery in Patel's paper is that aging actually limits the growth of the original primary tumor.
Wait, limits it?
It limits it, but, and this is a massive but, it drastically promotes the cancer's ability to spread or metastasize.
Wow.
The tumor is functionally uncoupling its local growth from its ability to invade other organs. It essentially stops expanding its primary footprint in the lung, and instead it prioritizes packing its bags and spreading everywhere else.
Okay, I want to really dig into that because that completely flips the script on how we view tumor growth.
It really does.
To understand how the team discovered this uncoupling, we need to look at how they fixed that age disconnect in the lab.
Right, the tools they used.
Yeah. They used a very specific, genetically engineered mouse model. Now, we aren't going to bog you down with the alphabet soup of the exact genetic sequencing, but this model is a staple in oncology. It's usually referred to as the KP model.
Right, and the power of this specific model is that these mice are engineered to develop lung tumors that perfectly recreate human lung adenocarcinoma.
Which is the most common type, right?
Exactly. And crucially, unlike some older lab models where the mice have no immune system so human cells can be implanted.
Right, the naked mice.
Yeah, the nude mice.
Right.
Unlike those, this KP model develops the cancer naturally within a fully intact functioning immune system and a normal cellular microenvironment.
Which is so important for seeing how it actually behaves.
Yeah.
And the researchers did something so simple, yet entirely brilliant. Instead of just running the experiment on young mice, they split their subjects into 2 distinct groups.
Such a basic idea, but so impactful.
Right. First, you have your young group, induced with tumors at 2 to 3 months old. Again, your standard lab-age teenager equivalent mice.
Yep.
But then you have the old group. They literally just waited. They let these mice live in the lab until they were 18 to 19 months old before triggering the cancer.
Which is a long time in a lab setting.
It's an eternity. When you map out that timeline, following the multi-month experiment, the older mice correspond perfectly to a 70-year-old human.
The exact peak demographic for non-small cell lung cancer.
Exactly.
And the comparison between the two groups was staggering. When they examined the lungs at the end of the experiment, the older mice actually had a 2.5-fold decrease in the overall burden of the primary lung tumor compared to the young mice.
2.5 times less.
Yes. When they looked at the tissue under a microscope, the tumors in the older lungs were fewer in number, they were physically smaller, and the cells were dividing at a much slower rate.
Okay, let's unpack this. Because if I'm a patient and my doctor tells me my tumor is smaller and growing slower, I am celebrating.
Naturally.
That sounds like a victory. Why is this paper presenting this as a terrifying realization about aging and cancer?
Because you have to look outside the lungs.
Oh. While that primary growth stalled out, the metastatic burden, meaning the spread of the cancer out into the lymph nodes and into distant vital organs, it skyrocketed in the older mice.
Oh, wow.
The tumors weren't growing in place, they were escaping. And just to be absolutely sure this wasn't some different type of cancer forming, they checked the specific cellular markers.
Right, to make sure it was apples to apples.
Exactly.
Oh.
Both the young and old tumors originated from the exact same type of lung cell, the alveolar type 2 cells. They were both positive for the pro-SBC marker.
So the starting line was identical.
Identical. But in the older environment, the cancer chose escape over local expansion.
Okay, I think this is a perfect spot for our first summary. Let's do a quick recap for you listening, kind of solidify these concepts before we move on.
Good idea.
So bullet point 1. Cancer research usually studies young mice, which completely misses the context of an aging immune system and environment.
Bullet point 2. When we actually use older mice, the 70-year-old equivalents, they grow smaller primary lung tumors, but they experience significantly more metastasis.
Spot on.
So here's a question for you to chew on as we move forward. Think about your own problem solving. When faced with a roadblock, do you try to push through it, or do you change your environment?
That's the real question.
The tumor, as we'll see, chooses the latter. What would it take for a cell to make that leap?
Well, if we connect this to the bigger picture, answering how an aging lung forces a tumor to stop growing and start spreading comes down to one core concept.
And that is?
Cellular stress. Aging and cancer actually share a lot of underlying biological traits.
Like what?
As we age, our cells slowly lose their ability to perform quality control on the proteins they build. It's called a loss of proteostasis. We also see changes in how cells sense and process nutrients, and we see massive shifts in epigenetics, how DNA is managed.
Let's try to visualize this. I always like to picture a single cell as a sprawling manufacturing plant, a cellular factory.
I like that analogy.
Its main job, especially if it's a tumor cell, is to constantly produce protein so it can build more cells and expand the tumor. But because the body housing this factory is 70 years old, the raw materials coming in are degraded, and the machinery on the assembly line is, well, it's getting faulty.
That is a highly accurate way to look at it. Proteins are like intricate origami. They have to be folded into very precise 3D shapes to function.
Right.
In this aging factory, misfolded proteins start piling up on the assembly line. They clump together. This creates a massive traffic jam inside the endoplasmic reticulum. That's the part of the cell responsible for protein production.
And a factory that advanced has to have an alarm system, right?
Yeah.
When the jam gets bad enough, it triggers something called the unfolded protein response, or the UPR.
Exactly. And within that response, the researchers found that these aged tumor cells become uniquely sensitive to one specific biological alarm system. It's a pathway controlled by a protein sensor called PERK. P-E-R-K.
I use a little mnemonic device for this. Think of Puck as the factory foreman who perks up the second he sees that massive pile of defective, misfolded proteins on the conveyor belt.
That's a great way to remember it. And when PERK perks up, it sounds the alarm, which activates a massive cell-wide protocol called the integrated stress response, or the ISR.
Okay, the ISR, what does that actually do?
Normally, the ISR is just a temporary pause button. It halts the factory's production line, stops making new proteins, and gives the cell's cleanup crew time to clear out the traffic jam.
Makes sense.
Once the jam is cleared, the alarm shuts off, and normal growth resumes.
But something goes horribly wrong with that off switch in the aged cells. And this is where the researchers looked at the epigenetics.
Yes, the packaging.
Right. For you listening, think of epigenetics not as the DNA code itself, but as the physical packaging of the DNA. Your DNA is incredibly long, so it has to be tightly spooled up inside the cell.
Like thread on a spool.
Exactly. Researchers use a technique called ATAC-seq to see which parts of the DNA are unspooled and open for business, and which parts are tightly packed away and closed. It's like wandering through a massive library and seeing which books are lying open on the tables and which are locked behind glass doors.
What they found when they looked at the library of the old tumor cells was profound. The DNA was wide open at the location for a specific gene called ATF4.
Okay, at F4.
But simultaneously, the chromatin, the DNA packaging, was physically locked, shut across a whole family of genes whose entire job is to resolve the stress. Things like HBA5, WFS1, GAD34.
Wait, so the genes that are supposed to act as the all-clear signal, the ones that tell the factory, hey, the jam is cleared, turn off the alarm, those genes are locked away.
Yes.
The factory managers are basically locked out of the control room.
Precisely. Because those resolving genes are permanently inaccessible, the cellular stress response never turns off. The cell enters a state of permanent chronic panic.
That sounds exhausting for the cell.
It is. And because that alarm is constantly ringing, it drives up the production of that open gene we mentioned, ATF4. In this aging environment, ATF4 becomes the absolute master commander.
The hub of the whole operation.
Exactly. It's the hub that receives this panic signal and tells the cell, this local environment is completely toxic and failing. We need to change our identity, abandon this tissue, and leave.
Let me pause and map this out. I want to make sure we have this fully synthesized, so let's do our second quick recap and bullet summary.
Go for it.
Bullet point 1. Aging causes protein stress, which triggers that PRARC factory alarm and activates the integrated stress response, the ISR.
Perfect.
Bullet point 2. Epigenetic changes, the locking of the library books, keep the stress resolution genes locked, creating a permanent panic state.
Yep.
And bullet point 3. This chronic stress state continuously elevates ATF4, which acts as the master commander of metastasis.
That covers it.
So here's the next question for you listening. If you were a tumor cell stuck in a permanently stressed environment, wouldn't you want to pack your bags and move? How exactly does ATF4 build the vehicle for that cellular escape?
That is the critical mechanical question the researchers had to answer. We know ATF4 is elevated, but physically, how does it permit cancer to spread?
Right.
It comes down to overcoming a biological failsafe called anoikis. A-N-O-I-K-I-S.
Anoikis. It sounds like a Greek tragedy, and, well, for a cell, it kind of is.
It really is.
Let me see if I have the analogy right here. Normal human cells are highly social, right?
Yeah.
They are physically anchored to their neighbors and to a structural scaffolding called the extracellular matrix.
That's their home.
Yeah. If I go to my garden and rip a plant completely out of the soil, it's going to die very quickly because it's detached from its life source. Anoikis is the cellular equivalent of that.
That's a perfect way to describe it.
If a normal cell, or even a young tumor cell, detaches from its local scaffolding and tries to float away into the bloodstream, it triggers an automatic self-destruct sequence.
Exactly. The bloodstream is an incredibly hostile environment for a cell that isn't designed to be there. But this is where the master commander, ATF4, exerts its power.
What does it do?
ATF4 grants the aged tumor cells anoikis resistance. It essentially builds a protective spacesuit for the cell.
A spacesuit?
Yeah. This spacesuit overrides the self-destruct sequence. It allows the cancer cell to survive detachment, survive the turbulent journey through the bloodstream, and eventually seed itself in a completely new, distant organ.
And what I appreciate about this paper is they didn't just theorize about this spacesuit. They proved it from multiple angles.
They were very thorough.
First, they used chemistry. They took old tumor cultures and treated them with a specific drug called ASRIB, ISRIB. It inhibits that stress alarm we talked about.
Right. They gave them 2 micromolar of ASRIB twice daily.
And by chemically shutting down the alarm, the ATF4 protein levels dropped. The cells lost their protective spacesuits, and their ability to metastasize in the mice completely plummeted.
They also proved it genetically, which is amazing. They used CRISPR-Cas9 technology.
The molecular scissors.
Yes. They acted like molecular scissors and literally snipped out the gene that creates ATF4. They also used a doxycycline-inducible shRNA just to be sure.
And what happened?
Without that gene, targets like ASNS and SLC7A11 dropped. Interestingly, 4EBP1 didn't change, but the metastatic advantage of the old cells vanished entirely. They were stuck in place.
Wow. But here is where my mind was completely blown reading the methodology. They did the reverse. They essentially played God.
The overexpression experiment.
Yes. What happens if you take a young tumor cell, one that normally just grows locally and never spreads, and you artificially force it to produce massive amounts of ATF4?
This was the ultimate test to see if ATF4 was just a bystander or the actual driver of the disease. When they artificially elevated ATF4 in young KP mice, and notably when they did it in human A549 lung adenocarcinoma cells grown in the lab, The results were explosive.
Explosive how?
It was entirely sufficient to induce massive metastasis. The young tumors instantly behaved exactly like old tumors. They underwent a physical transformation, epithelial mesenchymal transition, or EMT.
It became mobile.
Highly mobile, and they gained metabolic plasticity, to the point where they acquired the ability to colonize extremely unusual locations like the heart tissue.
Wait, they found lung cancer cells growing in the heart just by flipping this one stress switch?
Yes. The ATF4 protein gave them such immense metabolic flexibility and survival armor that they could thrive in environments that would normally destroy them. It proved undeniably that ATF4 is the engine of aging-induced metastasis.
That is just incredible. Okay, let's lock this in with our third recap.
That's good.
Bullet point 1. ATF4 grants Anoiki's resistance, acting like a spacesuit that lets cells survive detachment and travel through the blood.
Correct.
Bullet point 2. Blocking ATF4 with drugs like IceRib or snipping it out with CRISPR completely stops the metastasis.
It halts it in its tracks.
And bullet point 3. Artificially adding ATF4 to young, lazy tumors instantly makes them highly metastatic.
Exactly.
So here is the million-dollar question for our listeners. We've cured metastasis in aging mice, but what does this mean for the patients in the clinic right now? How does this change the way we look at human therapies?
To answer that, the researchers had to cross the bridge from the pristine lab environment to the incredibly messy reality of human biology.
Which is never easy.
No, it's not. They took these findings and validated them against massive, real-world human data sets. They analyzed a global cancer genome database called TCGA-UA, and they also looked at a very specific cohort of patients in Sweden who had surgically removed KRAS mutant lung tumors.
And this is where the hair on my arm stood up. The human data perfectly mirrored the mouse models.
It's rare that it aligns so perfectly.
They looked at the tumors and found that this ATF4 master protein is significantly higher in human patients over the age of 65 compared to those under 60. But the impact that protein has on actual human survival is a sheer cliff. I mean, in early stages, stage 1 and 2, ATF4 didn't change survival much. But in advanced stages, 3 and 4.
It was devastating. High ATF4 caused the median survival to plummet from 2.61 years down to 1.28 years.
That's cutting their remaining time in half.
The Swedish cohort data illustrates this even more brilliantly. When they looked at the overall 5-year survival rates for patients, those who had tumors with low levels of ATF4 had a survival rate of nearly 66%. Basically, 2/3 were surviving.
Okay.
But for patients whose tumors had high levels of ATF4, that number was effectively cut in half. Only about 32.6% survived.
And if you stretch that timeline out to 8 years, the cliff gets even steeper, doesn't it?
Much steeper.
Patients with low ATF4 still had a 60% survival rate. But for the high ATF4 patients, survival plummeted to less than 1 in 7, about 13.9%.
It transforms ATF4 from just an interesting lab observation into a robust, undeniable warning sign for poor outcomes in humans.
It really does. But within that grim data, there is a silver lining. Because the tumor relies so heavily on this ATF4 pathway to survive its journey, it creates a massive, actionable vulnerability.
Exactly. Building and maintaining that cellular spacesuit requires a tremendous amount of energy.
Makes sense.
The researchers discovered that the ATF4 axis forces the tumor to become highly dependent on one specific nutrient, glutamine.
Glutamine.
Yes. The tumor cannot maintain its metastatic state without consuming massive amounts of glutamine. It literally alters its metabolism.
And this is the aha moment of the entire deep dive for me.
Yeah.
When I read this, a huge mystery in the oncology world suddenly made sense.
The clinical trials.
Yes. For years, scientists have been developing drugs called glutaminase inhibitors, or GLS inhibitors. The goal was to starve tumors of this exact nutrient, glutamine. And when they tested these drugs in the preclinical trials on young mice, they worked brilliantly.
They looked incredibly promising.
But when they moved those exact same drugs into human clinical trials, they were largely a failure. They just didn't work.
It's a tragedy of the scientific process. But this paper explains exactly why it happened.
Because we tested those metabolic drugs on teenage mice. The drugs worked in the young lab environment. But when they gave them to the human patients in the clinical trials, the median age of those humans was 70.
Right.
The fundamental biology of the tumor had completely changed. The aging human body had reprogrammed the cancer to survive differently, and the drugs were suddenly useless.
It's astonishing.
The age disconnect we started this conversation with is the exact reason those clinical trials failed.
It is a profound and, frankly, somewhat humbling realization for the scientific community. By ignoring the physiological age of the host environment, we were designing and testing drugs for a biological state that simply didn't exist in the actual patients who needed them most.
This is a massive shift in how we understand the disease. Okay, let's do our final 5-minute recap and bullet summary here to tie the human element together.
Let's do it.
Bullet point 1. Human data confirms that ATF4 rises as we age, and it drastically cuts survival rates in advanced non-small cell lung cancer. It's exactly.
Point 2. The ATF4 pathway rewires the cancer cell to become addicted to glutamine for its energy.
Yep.
And bullet point 3, realizing that older tumors process nutrients differently finally explains why previous metabolic drugs, like those GLS inhibitors, failed in human clinical trials.
That's the perfect summary.
So a question for you listening to ponder: if the tumor's behavior is dictated by the physiological age of its environment, is it possible for a young person to have an old, high ATF4 tumor?
That brings us right to the ultimate synthesis of why this research matters so much.
Let's talk about it.
Historically, we have viewed aging as just a passive background canvas. We thought of cancer as merely a collection of genetic mutations that slowly accumulate over time, and the older you get, the more time you've had to collect those bad mutations.
Just bad luck over a long timeline.
Right, but this research proves that aging is not a passive canvas. Aging is an active, aggressive modifier.
It actively changes the game. Yes. The physical deterioration of the body, the protein traffic jams, the epigenetic locked doors actively reprograms the evolutionary trajectory of the tumor. The environment dictates whether the tumor stays put or spreads globally.
And if you're listening to this wondering how this changes the future of medicine for you or your family, it highlights an absolute urgent need for precision medicine. But precision medicine that factors in biological age, not just chronological age.
Exactly.
Right now, we look at the date of birth on your chart, and we look at the specific genetic mutations of your tumor. But this argues that we need highly tailored interventions for older patients right from the start, because we now know their tumors are molecularly primed to escape and spread early on.
We can't treat a 70-year-old's tumor with a 20-year-old's playbook.
We really can't. We have to treat the soil, not just the seed.
Exactly right.
Which brings me to a final provocative thought I want to leave you with today. While digging through the supplementary data of this paper, there was a fascinating detail buried in the human cohorts.
Oh, the subset data.
Yeah. There was a small subset of young human patients, people chronologically young, in their 40s or 50s, who also had tumors with extremely high ATF4 levels. And guess what? Their survival rates mirrored the 70-year-olds.
They faced the exact same devastating cliff.
Which strongly implies that this stress alarm, this ISR ATF4 axis, isn't strictly counting how many birthdays you've had.
No, it acts as a tumor intrinsic molecular clock.
Exactly. It is a direct measure of physiological age of the actual cellular stress and damage inside that specific tissue, regardless of what your driver's license says.
It's all about the cellular wear and tear.
So think about this. If we can learn to measure the physiological age of a tumor using this ATF4 marker, could we predict and chemically block metastasis before the cell ever builds its spacesuit?
It's the holy grail of oncology.
Could we turn an aggressive, spreading cancer back into a localized, manageable one? Think about that next time you hear a headline about a miraculous new cancer cure found in mice. Ask yourself, do they cure a 70-year-old or do they just cure a teenager?